Hello everyone, welcome to another topic of biochemistry genetics. Today we are going to discuss about autosomal trisomies. So in these autosomal trisomies, we are going to discuss mainly about the Down syndrome, which is the trisomy 21, the Edwards syndrome, that is trisomy 18, and Patau syndrome, that is trisomy 13. And along with that, we are going to discuss in brief about the trisomy 8 and trisomy 9 syndrome. So in this lecture, I am going to describe sequentially about the Down syndrome, Edwards syndrome, Patau syndrome, the trisomy 8 and trisomy 9 syndrome. Now let us discuss about the Down syndrome. The another name of Down syndrome we also called the trisomy 21. So Down syndrome is one of the most common chromosome abnormalities, and the three main types of cytogenetic abnormalities that result in the Down syndrome phenotype and their relative proportions are: it is trisomy 21, which accounts for the 95% of the Down syndrome; the Robertsonian translocation involving chromosome number 21 accounts for 3 to 4%, and trisomy 21 Mosaicism, which is the post-fertilization mitotic error, that is accountable for 1 to 2% of the Down syndrome phenotype and their diseases. So what is the commonest cause of the Down syndrome? So one of the most commonest cause of the Down syndrome is the Meiotic nondisjunction. So you should always remember about the Meiotic nondisjunction. So 90% of the case, the extra chromosome 21 originates always from the mother. So we always say that the Down syndrome, which is trisomy 21, increases in risks in advancing maternal age. So as the maternal age increases, like above 30 or above 35, then the chances of the Down syndrome increases, the risks of occurring of the Down syndrome increases. So now next let us talk about the findings in the Down syndrome. You can also see in this picture along with the findings. You can see the intellectual disability in the child, flat faces, prominent epicanthal folds, single palmar crease, incurved fifth finger, gap between first two toes. So while examining, we can also see the duodenal atresia, Hirschsprung disease, the congenital heart disease like atrial septal defect, and we can see the brush-filled spots in the child with Down syndrome or the person with the Down syndrome. So there is also the increased risk of AML and ALL, the acute myeloid leukemia and acute lymphoblastic leukemia. There is increased risk of AML and ALL in the child with Down syndrome. And there is correlation of the Alzheimer's disease and Down syndrome, that the early onset of the Alzheimer's disease is also related with the Down syndrome, that is the trisomy 21. And there are various screening can be done with to know the disease, if the child has disease, the Down syndrome or not. At the first trimester, we can check the beta HCG, the inhibin, and the pregnancy associated protein A. The pregnancy associated protein A we can see. So these are the three markers you can see, and we can analyze the child has Down syndrome or not. So in the Down syndrome, in the first trimester, in the USG in the first trimester, we can see the inclusive nuchal translucency and hypoplastic nasal bone. So in the Down syndrome, in the first trimester, we can see the increased nuchal translucency and hypoplastic nasal bone. And the other markers of the Down syndrome are the beta SAC. In the first trimester, the beta SAC level is increased. The level of inhibin is also increased, and the level of plasma pregnancy associated plasma protein is decreased. So at the last, I want you to know the 5 A's of the Down syndrome that is always associated with the Down syndrome. That is advanced maternal age, duodenal atresia, atrioventricular septal defect, early onset Alzheimer's disease, and increased risk of AML and ALL. So this is the brief explanation of the Down syndrome. I hope you liked it. This much for today. Thank you.